Assalamu alaikum. We are again here for online lecture. The subject which we are studying over here for MPhil semester 2, fermentation and its industrial applications. Lecture number 20 was going on, 19 was going on, and this is lecture number 20. And we'll be talking about the downstream processing. Downstream processing, a complete chapter in which we are using different techniques to isolate our desired product from ferment, from mixture. We have studied various techniques because there are several thousand type of reactions on industrial scale, and every fermentation type or every fine end product requires a unique setup of techniques to isolate, to purify, and to harvest the desired product. And most of the products which are produced by fermentation are very rare, expensive, and very fragile and very sensitive because most of them are used for human disease treatment. Sometimes these things are used for food, for animals, for plants. So if there will remain any impurity, it will lead various type of disasters and problems. So this is very important that every end product, whatsoever is that must be free from all types of contaminants. If you will sell, if you will launch it as it is in market, that will lead problem. So we have to go at least and at any cost, we have to remove all type of contaminants. So for this, several type of techniques has been developed. Advanced techniques also has been introduced to purify, to isolate, to separate, to purify, to polish your end product, because these will be used in market by human and by public. So uh, here in our previous class, just if you remember, we were talking that chromatography, and in chromatography there are various techniques. If you will see over here, this is affinity chromatography. So we will talk over here. Uh, this course is MIC531, lecture number 20. We are talking in downstream processing, and in downstream processing, we are talking about chromatography techniques, and various types of chromatography techniques are there, uh, uh, which are used at micro level to separate the molecules from each other. And, you remember my name is Mohammed, your instructor. Okay. So we will talk about chromatography technique. So what is affinity chromatography? Affinity chromatography separates the protein of interest on the basis of reversible interactions between it and its antibody coupled to chromatography beads. So the production of protein is very sensitive because production of protein is not an easy job. Various peptides are produced which are used for the different type of treatment, protein, antibodies, antigens, and many things are protein in nature. So when we are producing by fermentation a protein, that is very sensitive, very fragile, and very small molecule. So we have to use a sophisticated, advanced, and optimum technique to separate that protein from mixture. So affinity chromatography is a technique of one of them, a technique. affinity chromatography among them is one of the techniques. So we will see the principle how it is. Suppose this is column, okay? It is full of beads. This is bead, this is bead, this is bead, this is bead. Sometimes you are saying that these are resins, go small round substances which are made up of resin, which are filled in a column and packed in a proper way in compact form. That beads space between the beads will be very small, it will be negligible, and completely it must be packed. So proper technique is used. So for this, what we are doing, suppose these beads are coated by antigen. Suppose we produced a protein, an antibody for any antigen through fermentation. We produce an antibody which have nature protein for a particular type of antigen. We want to separate it from mixture, and we know very well that in mixture there are various type of substances are present. Waste material is there, water is there, additionally metabolites are there, secondary tertiary metabolites are there, waste material is there, bacterial dead bodies are there. So we have to separate all these things. Just we need our protein, which is our antibody. So what we will do here, we will use affinity chromatography. Affinity chromatography, separation of proteins on the basis of reversible interactions between it and its antibody, coupled to a chromatography bead here labeled antigen. We can see here. Okay, concentrate. This is column. You have this is bead which has been coated with this antigen. This is the antigen. This is your antibody, and this is antigen. A bead has been coated. Bead resin in column has been coated by antigen. Okay, has been coated by antigen, and it has been packed in column now. What happens now? Let's see. We will pour the solution which contains our protein, other waste material, and many things we don't know. But we are sure that this solution contains our desired product, which is antibody, which is antibody, and other molecules are also there. So what will happen when we will pass our solution from this column? Everything will go down, will move downward through gravitational force or through mobile phase. And a stationary phase contains beads plus your antigen. What will happen during this travel? The antibodies will bind with antigen. Antibodies will bind with antigen, which antigens which were coded over the bead, and antibody will bind with antigen, and all other molecules will pass on. Other molecules will pass on. Okay, we are successful here to separate our desired protein, which will be absorbed, which will be reversibly interact with antigen which were coated on bead. So what will be? With the high selectivity, high resolution, and high capacity for the protein of interest, purification level in the order of several thousand fold are achievable. So what will happen? What happened now? All things went down. only protein has been binded with these antigen, because these proteins are binded with antigen in reversible form. So now we will pour any pure solution uh, for the elution, which will remove these antibodies from antigen, and these antibodies will be collected in separate parts. So the protein of interest is collected in a purified, in concentrated form, because all things has been removed now, concentrated form. Biological interactions between the antigen and the antibody, which is protein in nature, can result from electrostatic interaction, van der Waals forces, and hydrogen bonds. These all bonds and interactions are reversible. That antibody, which is protein in nature, will bind with antigen with these interactions, electrostatic interactions, van der Waals forces, or hydrogen bonds. And to remove and to break down these bonds is very easy. To elute the protein of interest from the affinity beads. Now, beads have antigen plus our desired protein, which is antibody. It, it have only this thing. Okay. The interaction can be reversed by changing the pH or ion strength. When in mobile phase, we will change the pH of the mobile phase, and we will run it through the column. All protein, all antibodies which has been binded with antigen with above mentioned forces will come into the solution due to change of pH and will move down. The, the concentrating effect enables large volume to be processed, and by this method we can do a huge amount of volume we can process for this purpose. The protein of interest can be purified from high level of contaminating substance. The protein of interest can be purified from high level of contaminating. Can you understand? Making antibodies to the protein of interest is expensive. It's very sensitive, because this during this COVID, people are need antibodies against COVID, but we cannot do this thing right now because we don't know the nature and growth of virus is very difficult. So if some because it is changing, it is restricted. Therefore, we are unable to make any antibody through fermentation process. Making antibodies to the protein of interest is expensive. So affinity chromatography is the least economical choice for production of is the least economical choice. So affinity chromatography is the least economical choice for purification for separation of antibodies from a solution. So maybe you could understand little bit this thing. We will see once more. Affinity chromatography, separation of proteinous molecules on the basis of reversible interactions between it and its antibody or antigen coupled with chromatography beads. So next thing, this is another technique. This is also a type of chromatography, which is called size exclusion chromatography. So from here, we also can assume and we can understand this. This is again a column. This is again a column which separates molecules on the basis of their molecular size. So this is gel permeation filtration chromatography. This is called gel permeation filtration chromatography. We also can use if our our research is related to such type of technique, and this technique is suitable for separation of our desired product from any mixture. So we can use this technique, size exclusion chromatography. So for this we are using gel permeation chromatography, GPC, gel permeation chromatography. How it works? Separates molecules according to their size. This is gel permeation chromatography, which is called GPC. It separates molecules according to their size. We have to concentrate on this point. This size exclusion chromatography separates molecules on the basis of molecular size. Low resolution, it have resolution less resolution, and tertiary and quaternary structures can be formed from the fine product. So we can see our here. Okay. So this is column. This is column. It is filled with beads. Column is packed with beads. Okay. Column is filled with beads. But we concentrate here in this technique, which is very important to understand. If you will miss this point, you cannot understand size exclusion chromatography. The beads are porous. In column, the beads which are present inside of the column, these are porous. So what happens? Beads are packed in column, and we will pass any solution from upper side of the column with a stationary with a mobile phase. So what will be? These molecules, all molecules will move downward with mobile phase. But what will be? Small molecules will go inside of the pores of the beads. Is column, if you will see here. Okay, these are the beads. These are the beads, and the beads are porous. These have small pores. So what will be when we will pour any solution? The small molecules will go inside of the pores and will travel slowly, 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 because they will trap. They will be trapped in various places, and large molecules will come fast. So large molecules will go first outside, and small molecules will come later. If we know the size of our desired product, here we will be able to separate our desired product. Okay, when these are coming out from the column, what we will do? We will collect in first open drops, we will collect large molecules, 1p, then small molecules, small p, then small molecules, those in another open drop. So what will be? We will separate all these molecules which are present in solution on the basis of molecular size. So first will come out large molecular size, molecules having large size, they will come out first, and it will be followed. Then small, then small, then small, and most small molecules will come at last. If we know the molecular size of our desired product, we can collect uh, from this column our desired product, and through activity we also can find that our desired product is our here. This is also a technique, size exclusion chromatography, separation of molecules on the basis of separation of molecules on the basis of molecular size. So, through this technique, molecules having a large size, they will come out first, and the molecules having a small size, they will come out from column later and slowly. Here is also one another technique, reverse phase chromatography. reverse phase chromatography. Reverse phase chromatography is an elution procedure used in liquid. If anything is present in liquid, so we can use reverse phase chromatography for purification and for isolation. Chromatography in which the mobile phase is significantly more polar than the stationary phase. Chromatography in which the mobile phase is significantly more polar than the stationary phase. So first we will talk about this, the dipole-dipole intermolecular forces between the slightly positively charged end of one molecule to the negative end of the another are the same molecule. This is called polarity. Molecular polarity is dependent on the difference in electronegativity between atoms in a compound and the asymmetry of the compound structure. So again, we are going to this slide. This is, suppose this is column. Okay, what is this? Hydrophobic stationary phase. This is stationary phase, and this is hydrophobic proteins will bind to the stationary phase, and other hydrophilic proteins don't bind, and these will wash out. So this is called reverse phase chromatography. Oppositely charged molecules will bind with this, and free molecules will pass on. So HPLC is also a type of the chromatography. HPLC configuration, high performance liquid chromatography. How it works, we will see over here. Many of most of you have been to laboratory and you have seen HPLC, but how it works over here, we will see a demograph. It is a bottle which contains a reservoir. It contains mobile phase. It contains any solution, organic solution, ethanol, methanol, benzene, toluene, any mobile phase which will carry your product through the column. Uh, it is pumped. If you will see here, this is pumped. What it does, it takes a solution from here and it will proceed over here. This is column which contains beads. Different type of columns are present, and according your desired product, you are using the column. From literature, you can find. I think it was a technical issue. Let's see what happened. Sorry for this inconvenience. This is pump, and it is injected. Here you are injecting your uh, product, your protein. You are injecting over here, and what will be? Solution will come, mobile phase will come from here. It will mix with your product and it will take into the column. A column will separate according to its principle, which we discussed in our earlier slides, that either it will separate it on molecular size based, affinity based, or whatsoever based. Okay. Here it will detect that this type of molecule has been passed, and it will give a graph on a screen. Detector will detect that this type of molecules, okay, small molecules, when it will be detected by detector and it will show on the screen in form of P. Then another molecules will come from column. Detector will detect this molecule and will make a P according to the concentration of that. Okay, it will generate here. If you will see the machinic type of HPLC is here. Here you can put a reservoir. Here you are injecting your solution here, your desired product, uh, and the detector is monitor is here, and you can form graphs over here according to the passage of this solution. I hope little bit you could understand from these techniques. These techniques are basically used for the separation of fragile, small, and expensive biological molecules which have been produced through fermentation. Uh, and various techniques are used. And here I will mention and tell you that we must we have to select. We cannot use all type of techniques. Just we have to use technique which is suitable, which is good, and which is economically suitable for our commercial or laboratory activity. If any technique is not giving good results, so we should not use that technique. We should use that technique which has been recommended in literature for purification, for isolation and separation of compounds. I hope you could understand little bit about chromatography. Here we are going to end the chromatography session, and next session we will go for further techniques. Maybe you could understand little bit, and uh, whenever you will go through research articles and through book, you will find more information, and during interactive sessions we can get understand this scenario in clear way. I hope you will understand it, and have a nice time. Thank you very much.